Good morning, everybody. Welcome. Morning, everyone. Welcome to day 2 of the ICES ASC. So this morning, it's my absolute pleasure to welcome Dr. Cornelia Jaspers to the stage. Cornelia is a biological oceanographer by training, and she's developed impressive expertise on the population dynamics of gelatinous plankton. She studied and worked in Germany, the USA, and Denmark, where she's currently the leader of the Center for Gelatinous Plankton Ecology and Evolution at DTU Aqua. Since 2019, Cornelia has been representative for Denmark in the working group on introduction and transfers of marine organisms, as well as Denmark's representative for the working group on zooplankton ecology. Today, Cornelia's talk will bring us along through her research into both the anticipated and observed changes in gelatinous plankton, and together we can consider what this might mean for the future of our oceans. Cornelia's talk today will be the first in what we hope is the beginning of a long tradition of highlighting outstanding early career research on the ICES Keynote stage. On behalf of SEEKS and the entire ICES community, please join me to give Dr. Cornelia Jaspers a warm welcome.
Thanks, Amanda. Yeah, good morning. It's a great pleasure to see so many, and I still see there are some people dragging into the room. I would like to welcome you. And as I said, it's a great pleasure and honor to actually be given the keynote and talk about my research today. The talk will be about aspects in marine science that we basically ignore or lack to acknowledge. But these aspects actually have the potential to be of importance under, global change and might actually be a key turning point in our future ocean. And the elephant in the room I'm talking about is jellyfish. And I promise you that with this talk, you'll get some insights about, the paradigms that are out there, how global change might actually change jellyfish abundances, and what the interactions are between jellyfish and fish. So I would like to come back to Alan, who in the opening ceremony talked about big ideas, that we need big ideas. And I hope that with this talk I inspire you that we need to have a discussion in the zooplankton group, but in the entire ICES community that includes gelatinous zooplankton organisms. And I really hope that with this talk I can, push forward that people get jellies on the agenda and don't regard them as the, slimy organisms that you shouldn't care about. Good, now the pointer's not working. But before diving into the research, I would like to acknowledge my mentors and colleagues that actually supported me along the way, and the funding agencies that actually allowed me to work on gelatinous zooplankton all the way since my master's. And all these funding agencies actually allowed me to dive into different aspects of the ecology, but also the evolution of gelatinous zooplankton. And I'm very grateful to the Willum Foundation. As Amanda just pointed out, this foundation actually allowed me to jump from a postdoc position, back to DTU Aqua, where I conducted my PhD, and where I'm actually now leading a center for gelatinous zooplankton ecology and evolution. So today's talk will be structured in different sections, and I would like to start with the population development of native jellies. Why do I say native jellies? Because there are a lot of non-indigenous jellyfish species or gelatinous zooplankton organisms that are the real problem maker. And I'll draw on some examples from my own research to actually explain you why. And I'll explicitly focus on one comp jelly that we actually have used as a model organism to understand spread dynamics and population dispersal, selection pressures of non-native species in their new habitat. The next part will deal with global change impacts, and I know this is a huge topic and broadly, discussed and controversially debated. But I would like to pick one positive example where a gelatinous zooplankton organism could actually be a turning point on our food web dynamics and counteract the elongation in the food web length that is expected due to global change. So this might have actually important consequences for fisheries. And that's why I'm also dedicated one part of my talk to jellyfish-fish interaction. So what do we actually know, and what are the mechanistics that need to be understood in order to include jellyfish-fish interactions into ecosystem models? And then I'll close with a food for thoughts at the ICES community, and look forward to your kind of discussions. So when I talk about jellies, or people talk about jellies, it's basically an umbrella that's grouping a lot of different organisms that are everywhere in the animal tree of life. But to make life simple, I just stick to either jelly or gelatinous zooplankton. But they include members such as the true jellyfish, that sting and that no one likes. The comp jellies that have sticky cells and capture the prey by, basically glue. And we have the pelagic tunicates. You probably know salps, that can get in chain length longer than blue whales. But anyway, if one is looking across all these gelatinous zooplankton groups, they share a trait, and that's that they are transparent, they're mostly low in carbon content, but the most important thing is that they're a pain to work with. And that's why most people, hesitate to work with them, have ignored them, or just said that be careful that you don't slip on them in case, you have a jellyfish haul on deck. But they include world record holders, such as these giant Nomura of Japan that can attain biomasses of 220 kilogram. So you can imagine that these can be quite troublesome, especially to fisheries in Japan. As well as pelagic tunicates, here larvaceans, they're the same size like copepods, but these guys are amazing because they have generation times that are extremely short. So in the tropics, they actually complete an entire life cycle in 24 hours. So you can imagine that they have the potential to, as quickly as the algae, respond to increased food availability. Jellies have raised a lot of public attention because they form these blooms, and bloom and bust population cyclicity is part of their inherent, nature. But still, increasingly, we have seen that there are jellyfish blooms interactions with fisheries, such as here in Japan, where you see these giant Nomura, 200 kilogram in wet weight, drifting into gill nets and actually leading to severe interaction with fisheries. And these interactions have been picked up by high-flying journals, such as Nature as well, which highlights the attack of the bobs, so that we really have a problem. Current biology, you might know the paper by Chris Linham from the Miguela current, fishing down the food web, and jellies take over in heavily overfished ecosystems. But we also have a problem with deadly poisonous box jellyfish, for example, in Australia, that are range expanding, not only in Australian waters, but also to Thailand and negatively impacting tourism. So all these kind of, pictures and highlights have raised two paradigms. First of all, that jellies take over under global change, and second of all, that they're a dead end. But these paradigms need actually to be challenged because jellies have systematically been disregarded in most food web studies. Long-term data are scarce, and I'll show you an example of that. And basically, the mechanistic top-down control is unknown. So we have no mortality rates of different life stages of different jellyfish, groups. So do jellies take over under global change? To dive into this topic, I'll first look at the population development of native species, and then go further with non-indigenous to outline that these might actually be the ones that are the troublemakers. If you look at this world map with the different colors, this is actually one of the two studies out there that compiled all the world's records about jellyfish, population dynamics over time. And this paper by Lucas and Daniel Pauly, they actually include fuzzy records, just as fisheries and newspaper reports. And while you see that some areas show a high certainty of jellyfish blooms, and an increase in abundances, there are large areas where the increases are of low certainty. And there are also a lot of areas where there is no change or even a decrease. If one's now zooming into one example from the Bering Sea, you can see that there is a high fluctuation, and basically the biomass of jellyfish, here depicted on the y-axis, changes over orders of magnitude. And if I might quote Dave from his keynote yesterday, he said this snipe pipefish, the elongated seahorse fish, that one shows, large population fluctuations as well. And depending on where you actually snapshot this time series, you might end up with a conclusion that they're increasing or decreasing. So this really highlights that we need to have a mechanistic understanding. And what we know for sure is that non-indigenous gelatinous organisms are a problem. And this here is a small video sequence that I'm going to show. It was featured on German television 1.5 months ago. And it actually shows a rhizostome jellyfish, Rhopilema nomadica. It has been invasive in the Mediterranean Sea since the 1970s. It comes from the Indo-Pacific. It attains pretty high abundances, and it has actually been listed as one of the key concerned species of invasive species by the European Commission. And one jelly of this species can attain up to 10 kilograms in wet weight. So you can imagine the white bobs that you see on this image here. They represent quite a lot of gelatinous biomass. So you see the extent of this jellyfish bloom. It's not something that you just have in a cove or in a bay, due to currents drifting in. It's huge abundances that these Rhopilema nomadica can actually attain, and have been associated with severe impacts with cooling water intake, desalination plants in Israel, as well as tourism. A lot of people don't want to go swimming. I don't know why. But this is only one example. We also have another member, Blackfordia virginica. This guy doesn't get larger than this size. It's a hydromedusan jellyfish, and people think, it's so small, it has no impact. But it's actually expanding in brackish water and estuarine system across Europe, and it can attain huge abundances, again, up to 1 individual per liter, which is basically, one animal in the milk bottle that you take out of the fridge in the morning. And we have found this jellyfish in Kiel Canal for the first time in 2014 and confirmed it from the southwestern Baltic Sea in 2017. And because of the low salinity of the Baltic, it's actually a species of concern that will eventually, as we expect, make its way to the Baltic Sea. Another example is the sea walnut, or Miniopsis leidi. It's not a true jellyfish, it's a comp jelly, so it captures its prey with sticky cells. And here you see a sequence from our Danish waters during late summer. So all the small dots are jellies, different sizes from 1 to 20 millimeter. But the maximum size these guys can attain are roughly 15, 20, 100 millimeter, sorry. I've been working too much with these guys, so I can't recall the sizes anymore. No, but such a 100 millimeter animal actually produces up to 13,000 eggs per day, and it is a simultaneous hermaphrodite, and we have shown that up to 90% of the eggs itself produces are self-fertilized. So you can imagine that it's quite a potent invasive species. When I started my PhD in December 2008, nothing was known about the systematic range expansion of Miniopsis. And I'm very proud to actually present this map, which has been, pulled together with 47 co-authors, more than 12,000 presence-absence data points. And we can see that Miniopsis is present in large areas of northwestern Europe, but also in the Mediterranean Sea. Where does Miniopsis come from? It's native to the east coast of the US. And we can differentiate 2 populations, a southern one and a northern one. And the nice thing with Miniopsis is that it actually has a lot of background information, such as a fully sequenced genome. So this allows now for investigating spread dynamics on a scale that is unique for invasive species. So the southern animals actually got introduced to southern Europe, so the Black Sea, whereas the animals from the north got introduced to northwestern Europe. And we have resequenced 72 full genomes of Miniopsis from all invasive and native areas that you see depicted here in color code. And we could show that looking at the northern invasion, the animals are pretty similar. Whereas if you look at the southern invasion, there is a huge genetic difference between the source and the invasive range. Interestingly, the population that we have recently observed in the northwestern Baltic Mediterranean Sea also has a large differentiation compared to its native, to its Black Sea population. And now I'm going to challenge you, because these full genome data can actually be used to reconstruct the past. So what we see here is generations on the x-axis and effective population size on the y-axis. And we can actually see how the population fluctuation was throughout, the history of each individual. And I will show you a plot where you see different colors. Every line, every colored line represents the full genome of one individual. And the color responds to the map that you see on the left-hand side, where you have the different populations. So native south will be red, and invasive north will be purple. If one's looking into the past, one sees that all animals, irrespective of where they come from, they have one source, so they show ancestry. And we can also see that the lines depart. So basically, where colored lines depart shows you that something happens on population level. And this indicates here that this is the spread in the native range, where the southern and the northern population kind of expanded in different areas and showed a dramatic increase in the effective population size. But now zooming into the southern invasion, we can interestingly see that all animals actually stem from the southern native population. They show a large increase in the effective population size, meaning that we had a lot of animals that got transferred and they showed, a range expansion, positive population growth rate. And then in more recent times, we actually see a split between the Black Sea and the Western Mediterranean population, which is, consistent with that we had animals observed in northwestern Med Sea. But these data are the first proof that these animals come directly from the Black Sea as a secondary branch expansion. This could not be resolved before. And looking at the northern invasion, it's very interesting because we see that basically both lines very closely follow each other and that there is not a large increase in the effective population size. And this actually indicates that we have small but recurrent introductions from the native range. Whereas if we look into the southern invasion, we see, or we can reconstruct that the invasion into the Black Sea, where we have a large population differentiation and a large increase in the effective population size, is due to a large introduction event in the past, which basically stopped because we see that there's there is no gene flow anymore. And this observation that we have from this PSMC analysis actually fits also if you look at history. Because in the 1980s, we had a problem with the Cuba crisis. We similarly had a problem with oil and traffic activity with Russia. So the occurrence of Miniopsis in the Black Sea actually fits the former shipping routes between Cuba and the Black Sea. And when we look into the recent range expansion, it fits a classical secondary range expansion where you have a bottleneck population and a reduction in the effective population size. So this is just to illustrate that molecular data can actually help you to understand and unravel, the invasion dynamics that you have observed, that you could not observe in the past. But the striking question, if you look at this map of Miniopsis, in Eurasian water, is how come that they are present and thriving in the Black Sea and Caspian Sea, but they're absent from the Baltic Sea? Well, we did some reproduction experiments with acclimatized animals to different salinities, and we could actually show that at the low salinity that's characteristic for most of the Baltic Sea, there is basically no reproduction. But looking into another example, how come that Miniopsis actually forms hotspots in Dutch and Belgian warm sea waters, but is absent from close within water along the UK east coast? So actually in the UK, Miniopsis has only been confirmed from eDNA so far. No animals ever caught. Well, the physical oceanographers will say, of course it's ocean currents. But if you actually look into the invasion literature, ocean currents have not really been linked and considered in this context. So I was fortunate that some physical oceanographers joined the endeavor with our synthesis paper, and they reconstructed here the surface currents of the upper 50 meter in the North Sea over a period of 7 years. And the warmer the colors, the higher the stability of the ocean currents. And you can see that there is a very persistent, current flowing to the south along the UK coast. And this fits together with the observation that even though we have Miniopsis in these hotspot areas, they don't make their way up to the north. And then we were very lucky to actually have a natural experiment that allowed us to really pinpoint, so what is the contribution of ocean currents to the spread? Up to 2010, Miniopsis was widely distributed in Western Eurasia, I'm talking about the north now, in northwestern Europe, and you can see all the way up to Trondheim Fjord, which is roughly, 65 degrees north. But then we had a period of very cold, February, March temperatures in 2010 until 2013. And the second cold winter really kicked in and led to a disappearance of Miniopsis from large areas. But then, strikingly, one warm winter in 2014 was enough that the animals occupied the entire range that they occupied before. So we actually looked at molecular data, got some old samples from the central Baltic, where Miniopsis disappeared, and looked at the population structure with microsatellite before the cold sweep and after. And you can see that it's a totally different genetic makeup. So basically, the genotypes that are present in the Baltic before the cold sweep, they got extinct. And this clearly demonstrates that ocean currents are driving this spread. And they drive this spread because certain areas show favorable conditions, warm winter temperatures, for example. So these pockets that we have identified in the southwestern North Sea actually drive the recolonization of non-indigenous species. And I would just like to add to this story that we also looked at the population structure of Miniopsis throughout northwestern Europe in 2014. And basically, animals we caught in Belgium and all the way up in Norway in the Baltic, they were similar. So you couldn't differentiate them. And this indicates that it's one source population that was responsible for this drift. But what does this mean for the ISIS community? What do we do with this? If you look at this map carefully, you see that high weight southwestern North Sea, we have a lot of ports. We actually have among the largest ports in the world in this area. And they show a high interconnectivity where non-indigenous species can swiftly, recolonize large areas. So my suggestion would be that we really start to consider connectivity in invasion risk assessments. So it's not only vectors and habitat match that have been featured as the most important drivers of range and expansion of non-indigenous species. It's also ocean currents, the genetic makeup of the species, and its phenotype that have implications. So if we really want to halt species introductions in the long run, we need to have a holistic approach. And this is a sequence from Hornbeck, where I'm living, and you'll get some tuna, jumping tuna at the end of the talk, if you survive. But during late summer, we have a lot of Miniopsis in our waters. Here you see, again, a sequence, all these small dots, there are particles, detritus, but there are also a lot of Miniopsis larvae that are swimming around. And we were interested to investigate how come that Miniopsis is actually such a successful invasive species. And we posed the question if invasive populations in general are selected, meaning that they should, reproduce as early as possible. To look at this, we looked at laboratory-controlled cohort experiments. That means raising animals, from egg all the way to adulthood under at libitum food concentration. And if you do this in the native and the invasive range, you see the following. So size and egg production are related on a log-log scale. They have a linear relationship, as you would expect. But strikingly, if you look at the invasive range, you see that they mature earlier and that they have higher site-specific reproduction rates. And if you follow the white dots, you can see that this is independent of invasive populations. So the northern and the southern invasive population perform the same in terms of egg production. So this indicates a clear selection for earlier reproduction in the invasive range. And then we conducted a life history model. And with that one, we could actually also show that if you're optimizing for the population growth rate, you should mature as early as possible. So what we see in the invasive range. But in the native range is seldom are selected. I mean, in the native range, Miniopsis are in steady state. They're part of the ecosystem. So if we optimize for the lifetime reproductive success, we actually see that Miniopsis is expected or should mature at an intermediate size. But strikingly, what you should take home from this slide is that there is a relaxed selection pressure, over a large maturation size. So in the native range, where you likely have lifetime reproductive success being selected for, it doesn't matter for the animals if they mature at 10 or 40 milligram carbon. So invasive populations, yes, are selected. But what about jellies? Are jellies superior invaders? Well, with Miniopsis, we have shown that they mature at a smaller size and optimize population growth rate. But there are also other jellies that actually show unique life history traits. And I'll walk you through some of the examples. So true jellyfish have a life cycle. And I know a lot of fisheries biologists don't want to care about this, so that's why I have put up a life cycle of Miniopsis that we have just recently published. And you see that the adults, they are the guys that you see in the harbors and that you get out on the boat, but they actually produce small planula larvae that settle down on hard substrate, form polyps, and these polyps are pretty cool because they can clone themselves, but they can also strobulate. So that means it looks like a stack of Tellerken plates that you have in your kitchen when they are ready to release the small baby jellies, also called ephyra. So basically, one polyp during the asexual reproduction period can produce up to 25 baby jellies. But the important thing is that they can also form podocysts. So that means if you have a polyp and you throw them in anoxia, or you have really bad environmental conditions, they just form cysts. And these cysts can they can withstand a lot of, anoxia and really bad environmental conditions. They can survive for years. And then once the conditions are good again, they pop up. It's similar to diatoms or resting eggs of copepods. But these podocysts are really the key to understand population outbursts of jellyfish. And these podocysts can not only, form a new polyp that then eventually buds off baby jellies, they can also directly develop into medusa. On another occasion, so now we move back from the true jellyfish and go to the comp jellies. In 1982, it was suggested by a German zoologist, Chang, that ctenophores can actually reproduce as larvae. So before they metamorphose, they can also, reproduce. And he said that this should be a trait that allows them to really survive high predation pressure and all kinds of environmental conditions. And I would just quickly like to illustrate the life cycle of a comp jelly. So you have eggs, and then you have a small larvae. It goes through a growth stage where it's basically metamorphosing, and then it gets an adult. And what Chang observed in a lot of different ctenophore species, not only lobate ones, is that they can actually shortcut the life cycle. And in the Baltic Sea, and I like to highlight this example because it's from monitoring activities, so basically really boring monitoring, right? We found out that Mertensia ovum in the Baltic Sea is exclusively reproducing as larvae. So throughout 13 cruises, and I don't know how many thousands of cubic meters of water we analyzed, we never ever found a grown-up Mertensia ovum. And basically, experiments that we conducted on ship confirmed that these small larvae, even though they're still larvae, not more than 7 to 8 days old, they reproduce eggs. So this was the first time proof, on ecosystem level that this is actually an important mechanism and might explain why ctenophores are actually persistent in waters where you ask yourself, how can they survive here? So adaptation to survive unfavorable conditions, yes, gelatinous zooplankton have a lot of traits that allow them to do that, and we see that non-indigenous jellies are on the rise. So now going into the second topic, what about global change effects? And I know this is something that can be highly debated, but I would just like to pick one example from the Canadian Arctic by Lee et al., published in Science in 2009. You might say it's a bit old, but I think it's very nice to illustrate, because an increasing temperature along with a decreasing salinity has been shown to actually change the base of the food web. So here you see the nanoplankton, which is the large-sized phytoplankton that's important for copepods and for fish, the classical view. They decrease, whereas the smallest picophytoplankton, they're increasing. So even though the overall productivity stays the same, it's the smallest algae that thrive while the Arctic freshens and warms. And there are also other examples, and I would just like to present the niche model results from Flombaum and Marty that was published 2 years ago in Nature Geoscience, where they also showed that climate change impacts on picophytoplankton leads to a change in the biomass. And here you see, especially in the northern hemisphere, large areas in Greenlandic waters, for example, around the US, Canada, where you have an increase in the biomass of picophytoplankton. What does this mean? You increase picophytoplankton, you increase the importance of the microbial loop, and this directly relates to the fact that you are elongating the food chain. That means you burn off energy without, being transferred to higher trophic levels. So you decrease the transfer efficiency to ultimately fish, everyone cares about in this room. But what people tend to forget is that there's a shortcut in this food web. And this shortcut is actually that are the lean whales of the plankton, I call them. It's these larvaceans that have predator-prey ratios similar to baleen whales, like 10,000 to 1. So basically, they're the size of a copepod, and they feed a small, picophytoplankton 1 micrometer in size. It's crazy. But if you throw a net in the water and you count what you have, traditionally it's 90% copepods, and this 10% we don't care about, 90% is good enough. No, but it's this 10% that could actually make a change, because they can directly feed on the small side and shortcut the food web. And if you look at this plot, this is the specific growth rate, the specific growth rate temperature corrected against body mass. And if you compare both groups, you see that the red points are consistently above, but not only above, this is a log scale, significantly higher than the copepod growth. So meaning that these guys, they can really outgrow copepods and quickly respond to an increase in the picophytoplankton. And I'm going to show you and walk you through some examples where we actually have proven this on ecosystem level. So what's the importance of copepods versus larvaceans, so classical versus gelatinous prey? We crossed the Indian Ocean to actually have a transect where we look into highly productive ultra-oligotrophic and tropical waters. And what we can see is, only looking at the production, the copepod production is decreasing along the gradient. But the larvacean production is not only increasing, it's actually exceeding the copepod production by up to 5 times. So meaning that in certain areas of the Indian Ocean, ultra-oligotrophic waters, where you have the tuna that go there, there you actually have a gelatinous zooplankton group that's exceeding the copepod production. But if you look at abundance data over a large chlorophyll A gradient, you basically see that abundances never really peak up. So how come that you always have roughly 10 to 1,000 individuals per cubic meter and you never see these outbursts? Or you really never see them? Well, if you exclude predators, such as here done in mesocosm experiments, you see that, wow, suddenly the abundances are 2 to 3 orders of magnitude higher than what you normally observe. And similarly, when you have an ecosystem where a phytoplankton bloom happens due to, a river, rain event, river runoff-induced phytoplankton bloom, you also see that the abundances are drastically increasing. So this points to the fact that these guys are heavily top-down controlled. And there's actually a very nice review hidden in a book by Jennifer Purcell. And she has gone through the very, very old literature and found that there are more than 200 fish species, that feed on these guys. So maybe they need consideration, right? But what about the jellyfish-fish interactions? Are jellies a dead end? Well, the results I just presented point to the fact that this might not be 100% the case. But there is actually very interesting work, and this is one of the very, very few studies where you have laboratory-controlled feeding experiments by Jeff Runge from '87. And he confirmed from laboratory experiments that mackerel feed on Aglanta, a jellyfish, very common, in the North Sea, for example. So laboratory experiments confirm jelly feeding. But to get to a mechanistic understanding, we need more of these kind of experiments, 30 years old data should be, expanded to other species. But if you look at another study here from colleagues from South America of Argentina, this map that you see here is based on 69,000 fish that these Argentinian colleagues, analyzed, not alive, but fresh on the boat. And they found that 24 different fish species actually feed on ctenophores, and also to pretty high, extents. So you see the frequency displayed in different bubble sizes. And some of these fish species actually have a pretty high frequency of ctenophores in their gut diets. And similarly, Basti Huber, a colleague from DTU in the Baltic Sea, has shown that occasionally our cod also feed on the invasive Miniopsis. So gut analysis, fresh gut analysis are also important to confirm jelly feeding. But who wants to do that? A different way to approach the problem is through molecular tools. And we did this in the Sargasso Sea looking at eel larvae. And we found that more than 75% of their gut contents actually comprise of hydrozoans. And not enough, we also, of course, quantified all the target hydrozoans in the area, so small jellyfish. And we found that the abundances of the jelly actually match the calculated energetic demand of eel larvae. So this might be a key to culture eel larvae in captivity, which has still not been successfully. The life cycle of European eel has not successfully been closed in the lab. So molecular analysis confirmed jelly feeding. And now the last example, bluefin tuna, smaller 100 centimeters, they also feed on salps and jellyfish based on stable isotope analysis. And now people will say, yeah, it's certain fish species, it's all the larvae and the small size classes. But a recent paper from Günther in 2021 actually confirmed with molecular tools that larger tunas, 15 to 60 kilos, so not the very large ones, but they also feed on jellies, up to 27% of the gut content comprised of gelatinous prey. And now I would just like to show you the following video. So this is jumping tuna. It's not the Mediterranean Sea. This is Denmark. It's from a tune safari, so tuna safari off Helsingør, the Hamlet Castle, and I can highly recommend visit and have a look. So it's these really magnificent animals that are extremely fast and that were regarded to, be unable to feed on gelatinous prey because they have such a high energy demand. They need to be streamlined. They need to be fast. You see how fast they are. They're chasing a mackerel, swarm here. But also these guys do feed on jellies. And why is this important? It is important because fish exert a top-down control of jellyfish. But if we do not know what these interactions are and how large this top-down control is, we have no idea what's going to happen with the jellyfish populations if we overfish certain species groups. So there we could have used some sound. You hear the people screaming, yeah! But there's also another interaction. So now we have seen, we also know that jellies, of course, feed on fish larvae, for example. So there is the top-down control that jellies exert on the fish recruits. And we have shown this for early life stages of Baltic cod in the Baltic Sea. And here you see a Miniopsis, that's slowly entraining this invisible feeding current, a small, yard sac larvae of Baltic cod, and this is basically dead. But the thing is that it's only a very limited period of time that these fish larvae are susceptible to Miniopsis predation. It's only up to the first 4 or 5 days. Afterwards, when the yard sac is used up, the fish engage in swimming and they're too fast, so they escape Miniopsis. Miniopsis is only a threat to the very, very early life stages. But people feared that, especially this invasive jellyfish has a huge impact on the Baltic cod population because they occur in the same depth layer like the Baltic cod eggs. They sink down and accumulate, at the interface between low saline and high saline waters in the central Baltic Sea. But our experiments actually showed that these guys do not feed on the cod eggs. How come? An animal that's feeding with sticky cells is not feeding on cod eggs. That's really strange. And people in the jelly community did not like these videos that you're seeing there because it was destroying their argument. But we also looked from a more mechanistic perspective. So this is a 3D image. So basically you see the jelly and the mirrored jelly from 2 different perception or angles. And what you see is that the red-colored cod egg is basically passing the lobes, the inside of the lobes, where you have all the sticky cells. If this egg would have been a copepod, 99% chance that it would be dead. So this clearly shows that cod eggs are not retained by Miniopsis. And if you're interested in this, we can have a beer, and I can also explain you why, because we found out when I was over in the US with some colleagues. But it's not only us who found feeding of jelly is not significant on the eggs. It's also other colleagues from the Alfred Wegener Institute that have shown for the North Sea that Miniopsis is not feeding on fish eggs. And this is not only, cod eggs, they also looked at other eggs. So the take-home message from this section is that we really need to have a mechanistic understanding, because there are some interactions that are not the same between species, but we also really need to change this paradigm, because the increase of jellyfish population could be driven by the fact that we just remove, the lid. We remove the right predators, and then we create our own problem. But in order to actually address this, we need laboratory experiments. And this is a real challenge, and I hope that there are a lot of young people that would like to engage into this endeavor, because it's rewarding, but it's also a lot of work. So I hope that you're convinced that jellies are not necessarily a dead end in the food web, but that there are this top and bottom up, top down and bottom up effects that are really important to be addressed. So as a summary, the contributions: no dead end paradigm, we just heard this. There is a swift adaptation we have seen for Miniopsis. We have also seen some examples that there are gelatinous non-indigenous species that are on a rise. Most importantly, there are certain jelly groups that can actually exceed copepod production, and this is not consistently, included into ecosystem models. So this really is the key that should be, focused on in the future, because larvations could have large food web implications. And I only talked about the higher trophic levels. They also produce houses and are important in carbon sequestration, but that's another story. So coming to the last section, food for thought. I hope that with these examples, I kind of, grabbed your attention that there's more to the story, that we have the jelly paradigms, that jellies take over or not, and that we really need, as an ICES community now, to push the field and include gelatinous zooplankton into our zooplankton activities, but also on a larger scale. And an example to achieve this is from our American colleagues here, Rick Baudeur from NOAA and Mary Beth Decker from Yale, where they have included, jellyfish into bottom trawl surveys. This is a time series pretty kind of unique from the 1980s all the way up to now. A lot of people will now argue, yeah, that's nice. Yes, and we actually also know how to do it, because there are other colleagues from Cephas and Yves Romer that have written a very nice marine policy, statement outlining how you can cost effectively include jellies into ongoing activities. But a lot of people will still say, we don't have the resources, this is far too expensive. Yes, but if we want to understand ecosystem changes due to global change, these are key players that we should not ignore anymore. An alternative and the cheap way how one could get started without allocating a lot of resources would be to just include the number of fish trawls that couldn't be completed because they're overfilled with jellies. And this is an example from Limfjorden. Follow the red line, and you see the number of fish trawls that could not be completed, so nearly up to 60% in certain years in Limfjorden, where the trawls were unsuccessful due to jellyfish. So it's a quick and dirty rough estimate, but at least it's better than nothing. So in order to arrive at a systematic recognition of jellies, we need to change the mindset. And that's why I'm very pleased that young people and ICES is also pushing the young people to actually move into an area where they're open-minded and leave current paradigms. As I've just outlined, it would be really important to include jellies into the macro jellies and fisheries surveys. What we have done at DTU Aqua since 2014 is to include micro jellies into ichthyoplankton surveys. And I have colleagues in Sweden and in Norway that are doing similar things, but I think we should move forward to really coordinate this. And ICES is the right spot to actually discuss this and to move forward. So I think this is the perfect match. And I really hope that we can get some discussions going and that you leave this room and have the perception that, okay, there's a bit more to the story that jellyfish are at dead end and increase due to global change.
Thank you, Cornelia, for that fantastic talk. We have roughly 10 minutes for questions and discussion. So are there any questions from the floor? Yeah, there's one straight away in the middle. Yetta's on her way.
Thank you very much. I'm Aqua, chair of the working group on ballast and other ship vectors. We met yesterday, and first of all, I would like to congratulate you. I think it was an amazing talk. I think you're spot on with the harmfulness potential jellies. And if you want, I would like to make you the newest chair invited member of our working group. But I also have a question. In the Baltic Sea non-indigenous species expert community, we looked at a lot of different non-indigenous species, also jellies, but not just jellies. And when you look at these different things, such as population dynamics, environmental tolerances, habitat preferences, and you may find some connections here and there to try to find which species are going to spread or distribute different areas, and not only spread, but also have an impact. And you realize that people cannot predict the damn weather for more than 2 days reliably. So I guess, to what extent could you predict other jellies than Nemesiopsis to spread into new areas and having a significant impact? Thank you.
Yes, thank you very much for the very interesting questions. Of course, the example with the North Sea, where we document the conveyor belt, invasion dynamics, is very easy to pinpoint because it has such a huge magnitude, right? The Baltic Sea is much more difficult. But in terms of invasive species, invasive jellies that we can expect, Blackfordia virginica, for example, has this potential that it thrives in estuarine and low saline waters. So basically, it just needs to overcome, the southwestern Baltic. And once you have ship traffic or ship hole fouling that's transporting Blackfordia towards Finnish waters, for example, you'll have the problem. Another example, can I still use the slides? No. Good. Another example is if you recall, and now it's messy because I didn't know where it is. Okay, maybe that was a mistake. But okay, here, this map. So if you look at the entire European map, up here in the upper right-hand corner, you see that you have this large-scale connectivity pattern. And for Miniopsis, we have shown that they cannot reproduce in the Baltic. So basically, salinity limits their range expansion. But they're gradually actually moving their way forward. And we have now also detected them in the Bay of Biscay. So colleagues have shown that they are present there in eutrophicated areas. So basically, they can also move their way, against the currents. But if you go to the Portuguese coast, for example, the Atlantic coast, Miniopsis is very likely not to be able to establish there because of, the high turbulence and because of the oceanographic character of the region and the limited connectivity. So basically, there is not a large inoculum that could seed animals that they could successfully, establish there. The same is what we see in the English Channel. Because of the high tidal amplitude, Miniopsis has no chance to really establish, in these high turbulent areas. And we can also discuss why later on. But in terms of new invasive species that we are expecting, Rupilema nomadica, for example, if we get this jellyfish, which also has polyps, into northern European waters, I think we'll get a severe problem, especially in the Wadden Sea. So the ports really need to get consideration, and this is especially true for the southwestern North Sea that also shows very warm winter temperatures compared to other areas. Thank you. I hope I answered your question.
Thank you, Aqua and Cornelia. There's another question at the back here.
Yeah, thank you very much. That was really interesting. We're not all purely into fish. We think about other things as well. And that is really nice to see the ecological steps. I love ecology, it's the way to go. A question, you were looking at long-term historic records. I wondered if you'd looked at the Alistair Hardy Foundation SAFOS constant plankton recorder data sets. Slightly different jellies, I know, but there's a long-term, series time set, and I just wondered if it had played into your research at all. Thank you.
Yes, so I decided against showing you another plot, which actually includes this CPR data, which is this one. And now I'm badly preferred, or I need glasses. So it's the North Atlantic time series that you see, the second one on the bottom. Sorry. This is the CPR data. And basically what you see there is that there is no change in jelly abundances. But basically one also needs to consider that the whole of the sampling device is like this. So basically they're counting nematocysts, so stinging cells, and relating that to jellyfish abundances. So maybe not the right proxy to resolve that. Yes, thank you.
I have another one down here.
Hi, Cornelia. Thanks for your great talk. I have a question in regards to the North Sea example you were showing. So do you think that open ocean large-scale dynamics has an impact on that story you were, I'm thinking like Sopologyre. Do you think it has an impact on how the currents affect the species?
I mean, if you move to the polar ecosystem, the map by Flohnbaum, the niche model showed that you'll have a higher importance of picophytoplankton also in those waters. So basically, it's expected that larvaceans have a higher role in those waters. So that's definitely to be considered. Another story is that we actually have salps that are range expanding along the US coast. And we have now salt blooms off Alaska that we have never, ever had before. And that comes to the next question. So why are there no salps in the Arctic so far? So there are some, dynamics and potential invasive species that really need to be considered. But we, first of all, need to start looking. Because if we do not systematically look, it's too late. The train has already, started to go. So it's now that we really need to jump on and consistently include jellies in the different ecosystems.
There's another question here in the middle.
Thank you for a wonderful talk. I'm curious about the energy content. You say they're feeding, your observations of fish feeding on the jellyfish, but how much would they need to eat to get enough the energy that they need for to be counted in the food web and that?
So that's the unique thing about Jeff Runge in 1987, because he did laboratory experiments, right? And he showed that, okay, one Atlanta is actually comparable to 7 copepods because it's much larger, right, in terms of energy content. But Atlanta is also a pretty rigid jelly. So you need to consider that there are different jellyfish groups, and they not all have a low carbon content throughout their life. They are basically like a balloon. So you start out with a high carbon content, and then you blow up the balloon, and you actually, dilute your carbon content, increase your surface area, and thereby decrease your predation risk. And if you actually look on the carbon content, fish and jellies have the same feeding rates. But this is getting off the record. Basically, the take-home message is that jellies, they do have carbon, so they're not junk food, and they're easily digested. So there's a trade-off. And one is actually now also moving into the field that jellyfish are used for collagen production, because they have some special collagen structures that are, even important for food health, maybe. So that's where, the research is also going.
Is there another one? There's no questions online yet. But...
There's another.
I'll make it brief. You've focused on lateral patterns for, I think, very obvious reasons. I was wondering if you could comment on vertical patterns.
Yes. The three-dimensional nature of the ocean complicates everything. Yes, so in that context, it's important to consider, the growing extent of oxygen minimum zones, for example, because that's structuring the, three-dimensional ocean habitat. And there we have shown that hydrozoan jellyfish, for example, they're much more tolerant towards low oxygen waters. They seek refuge in low oxygen waters to actually avoid predation. So yes, there's also vertical structure, but it's too much to, expand into this talk.
There was one last question at the back. Someone raised their hands. Oh. The microphone's coming.
Thank you for a very interesting talk. I'm just wondering, did we already let the genie out of the bottle? I'm thinking about the chips also operate inland waterways. So I'm kind of wondering that should we also be concerned about those? Possibly as a route for connecting two very different sea areas, but also thinking about these inland waters. Of course, there's a difference in salinity and seawater, but do we have to be concerned also about inland waterways and shipping?
Yeah, so the thing is that jellies are primarily, saline species, but there are also certain jellyfish groups that can thrive in freshwater. We have the freshwater jellies. But if I understand your question correct, and I could draw on an example from Egypt, because they have these saltwater ponds where they basically raise fish. What they do is they catch plankton offshore and bring it with big trucks to the lakes. So they have imported their own problem, and got Miniopsis in these ponds. And they have severe problems because they are feeding on all the zooplankton and the fish are starving. So yes, I mean, shipping and other vectors, aquaculture, do have a potential impact on the spread of jellies, definitely.
There was one question from online, so for the hybrid format, I may ask that on their behalf. So it's a simple one, I think. Is the lack of predation on fish eggs limited to Miniopsis, or is it common to most jellyfish?
That needs investigation. As simple as that.
Okay, I think that's a good place to leave it then. Thank you once more, Cornelia. Thank you, everybody, and enjoy the rest of the day.